Okay, so today my topic is neoplasia or carcinogenesis. Neoplasia is basically a tissue growth that is unregulated, irreversible, and monoclonal. So basically, a cancer cell is not regulated. If we see a normal cell, so a normal cell under a positive stimulus undergoes into cell division and the cell cycle stops here. But the cancer cell, it divides not under a positive stimulus. So there is no stimulus coming for the cancer cell and it is continuously dividing. The cell cycle is not stopping. So this is unregulated and irreversible. Monoclonal means that all these cancer cells are derived from a single parent cell. So this is the single parent cell. Now, if a cell really wants to divide, so for division, there are two genes for it. That is the proto gene and the tumor suppressor gene. From proto, we get the word producing. So it means that if proto gene is stimulated, so the cell cycle will be there or the cell is going to divide. So the proto gene is the producing gene. However, the tumor suppressor gene is the suppressing gene. Means if it is activated, the tumor cell suppressor gene is activated, so the cell cycle is going to stop. Now, if I've drawn proto gene, the DNA, and the tumor suppressor gene. If this cell wants to have cancer, so it means that the proto gene should be stimulated so that the DNA gets activated and it undergoes into many cell divisions. And the tumor suppressor gene, which are the breaker genes that gives the negative signals to the DNA and stops the cell cycle, this should be mutated. Its function should be less. So the tumor suppressor gene function should be less and the proto gene, which is the producing gene, function should be more. Basically, in this video, we will discuss the proto gene in detail. Now, the proto gene is not like this simple one gene and producing many cells. It is basically a set of 5 genes altogether. So, these sets of 5 genes are here. Number 1 gene of the proto gene is the growth factor. Number 2 is the growth factor receptor. Number 3 is signal transducer. Number 4 is nuclear receptor. Number 5 is cell cycle regulators. So it means that the first and the second gene are the growth factor and the growth factor receptor. If the first gene is activated or stimulated, so it makes growth factors. Example is platelet derived growth factor. And if the second gene is activated, so it makes more and more growth factor receptors. Now in the cell, if the growth factor comes and binds with the growth factor receptor, so the growth factor receptor is stimulated. And this stimulated growth factor receptor activates the proto genes and this in turn gives positive signals to the DNA to make more and more cells. Again, this is going to lead to cancer if it's defected. So, this is gene number 1 and 2. Coming towards gene number 3, that is signal transducer. The example is RAS and ABL gene. Coming towards the RAS gene. As I said that if the growth factor receptor is activated, so under this growth factor, there is a very special type of protein known as RAS. Here this is RAS and under this RAS or in the mouth of the RAS we have a special protein that is known as GDP. Now if the growth factor receptor is activated, so this activation converts the GTP into GTP and this GTP in turn activates the signal transducers and gives positive signals to the DNA to make more and more cells. Now, if in a normal cell, if we see the GTP should be inhibited or should immediately be converted into GTP. so that the cells, more and more cells should not be made. Therefore, there is a special protein. So there is a special protein that immediately converts this GTP back into GDP. That protein is known as GTPase. So in the cancer cell, this GTPase is mutated. So here the GTPase is mutated. Therefore, there is increased cell synthesis and that leads to cancer cell. Now coming towards the gene number 5, that is cell cycle regulators. Now here if we see that there is the DNA, and just behind this DNA we have a very special key known as E2F. This key is like this, and this is behind DNA, and this key is basically under the mouth of the dog, and this dog is known as RB protein, which is holding this key backwards. If this dog open its mouth and the E2F is released, what happens is that this E2F binds with DNA, and this binding causes the DNA activation and the cell cycle undergoes. So if the cell cycle really wants to, if the cell wants to make more and more cells, so the dog mouth or the RB protein mouth should be open. In order to open the mouth, the RB protein asks for phosphates, and these phosphates are basically provided by gene number 5. So a man comes in the cycle and provides bag of phosphates. So here is the bag of phosphates which is providing to RB protein. This man with cycle is known as cyclin D1 gene, or CDK4. So again the mouth of the dog is open, the E2F is released, it activates the DNA and the cell undergoes into cell cycle. If this RB protein mouth is always open or it is defected or mutated, again it is going to lead to cancer. This RB protein is known as retinoblastoma protein, which is involved in retinoblastoma. Now coming towards gene number 4. Gene number 4 is known as nuclear regulators. Always remember the nuclear regulators are MYC gene. Knuckle is known as mimic karna. So nuclear regulators are MYC genes. So we have 3 kinds of MYC genes: C-MYC, N-MYC and L-MYC. The C-MYC we always see Burkitt. Okay, so if C-MYC is mutated, so that is going to lead to Burkitt lymphoma. If N-MYC is mutated, so that is going to lead to neuroblastoma, N for neuroblastoma. And if L is mutated, so L for lung carcinoma, small cell carcinoma. Basically, we will discuss in detail about the C-MYC. Now, where is the C-MYC? The C-MYC is present in chromosome number 8. And this C-MYC is always sleeping in this position. Because if it's activated, so the cell is going to go under cell cycle, or it is going to divide. So, and in chromosome number 14, we see a special IGH location. This IGH in chromosome number 14 is always activated because the body needs so much IGH always. Therefore, it is hyperactivated. Now, if we see that if there is translocation between 8 to 14, that is, if the MYC gene comes from chromosome number 8 and goes to chromosome number 14, so here the thing is going to look like this. And now in this chromosome number 14, the MYC gene will be hyperactive. So now the MYC gene will make more and more cells, and these cells will be very aggressive, and this is going to lead to Burkitt lymphoma. So here in 8 to 14 translocation of MYC gene of the C-MYC, the cells overproliferate and become aggressive. If we see the slide, so there are many cells here, and obviously in cell cycle, because they are going so fast, so there will be necrosis as well. So these are the necrosed area, and these are the cells which are replicating. So here there will be dark sky, starry sky appearance, because in the darkness here we see stars like necrosis. Okay, so there is stars like necrosis here, and we have many cells over proliferating. Now here we have the examples of POG. Just sum it up. I said growth factor receptor example is platelet derived growth factor. Growth factor is platelet derived growth factor. Growth factor receptor is HER2/neu, which is responsible for breast cancer if this is mutated, the RET and the KIT gene. S signal transducer example is RAS and ABL. Nuclear receptor is cMyc, nMyc, lMyc. CCR or the cell cycle regulators are cyclin D1 gene or CDK4. So that is all about proto-oncogenes. Inshallah, my next video will be on tumor suppressor gene. Do like my video if you have understand and subscribe my channel. Till then, Allah Hafiz.